Hi, I am Donalyn Dogin, and I'm going to present on my final year project, Mode 2, which is Research Review, entitled Entomopathogenic Fungi as Biological Control in Agriculture. The use of living organisms to manage pests is called biological control. So, agrochemical crop protection has improved crop quality and quantity worldwide. Entomopathogenic fungi, which is also known as the EPF, are a type of soil-dwelling microbe that uses cuticle penetration to infect and kill insects and other arthropods. Even though EPF have recently been commercialized, their broad potential applications have yet to be completely studied. Agriculture supplies most of the world's food and textiles. Agrochemicals residues can harm future crops and even humans. So their widespread and sometimes reckless usage has resulted in pest resistance, secondary pest reappearance, and disruption or destruction of natural enemy complexes, reducing the efficacy of natural control methods. Therefore, it is critical to investigate the EPF application as biological control in agriculture, their interaction with insects, efficacy as genetically engineered EPF, as well as the climate change's effects on the EPF. The objectives of this study is to review the application of entomopathogenic fungi as biological control in agriculture, to review the interactions of insects and fungi, to review the efficacy of genetically engineered entomopathogenic fungi as biological control, and to review the effects of climate change on entomopathogenic fungi. Now move on to the main content of the study. So one of the applications of EPF is that they can act as insect population regulators. Fungi-induced epizootics control nuisance insect population without the use of pesticides. For instance, aphids have a high infection rate. The EPF, known as the Neozygites frasini, has infected cotton-growing Aphis gossypi in Arkansas. The epizootics were common, and the incidence may have exceeded 90%. Another example of EPF is Entomophagary species, so they attack acrodids globally. In rainy or humid conditions, they are known to cause epizootics. EPF can also act as an endophyte. So an endophyte can live inside a plant without affecting its growth. So a meta-analysis by Rowe found out that the endophytes could combat drought, salt, and nitrogen deficit in agriculture. So this is supported by the findings of Chiang and Farias, where the Alternaria alternata, Paecilomyces formosus, and the Penicillium funiculosum can improve plants' resistance to drought, as shown in Table 1. Entomopathogens applications requires a suitable method to enhance the infection process. So there are several methods of inoculation which can enhance infections. This table shows some of the examples of inoculation method of EPF to plants. So some of the inoculation methods used are stick treatment, spraying, as well as soil drench. Move on to the insect-fungal interaction. So this figure shows the invasion of EPF in an insect. So the main modes of action that are used by EPF include the attachment of conidia, germination and apresorium, penetration, and the generation of secondary infectious conidia. Genetically engineered EPF can increase virulence. The three methods used are by utilizing pathogens' own genes, utilizing insects' proteins, and by utilizing the genes from other insect pathogens and insect predators. So from this table, it can be noted that the Beauveria bassiana, Metarhizium acridum, and Metarhizium anisopliae are the most common EPF used for the virulence enhancement. Next, abiotic stress tolerance can also be enhanced by the genetically engineered EPF. So the inconsistent performance of mycoinsecticides in the field are limited by abiotic stresses such as UV radiation, high temperature, and low water activity. So selecting the best growth substrates and conditions for conidia production can increase tolerance to these abiotic stresses. So this table shows the two factors which can affect the abiotic stress. So they are the UV radiation tolerance and the heat tolerance. Previous research have shown that the climate change can affect EPF. So according to earlier research, the Hippocrates ideal temperature for the efficient growth of the majority of EPF was around 30 degrees Celsius. So findings by Kamga stated that the desert locust susceptibility to infection and mortality from EPF, known as the Metarhizium acridum, is the best at 28 degrees Celsius. So Kamga agreed to a certain level based on the earlier research. Class stated that the high temperature, which is above 35 degrees Celsius, is a limiting factor for the performance of EPF, isolates Metarhizium anisopliae and the Entomophaga. So this supports the findings obtained by Borstate and Magan, where they found that only one strain of the Biovira bassiana and Metarhizium anisopliae was able to grow at a temperature of 35 to 37 degrees Celsius. And he added that no strain of Eiseria farinosa was able to grow at that temperature. For the significance of study, this study will be very beneficial in the agricultural field. The use of biological control to control pests can lower the usage of chemicals and cause less damage as well as contamination to the environment. By emphasizing the benefits of biological control, we can increase the public's understanding of its importance for a better health. To conclude, researchers and other institutions have conducted various large-scale investigations on insect-toxic fungi since discovering that they may help battle insects in agriculture. Fungal entomopathogens govern insect populations, which is crucial to environmental network self-regulations. So, the research gap of the study is that most studies mainly focus on the Biovira species as well as the Metarhizium species, but there is a lack of information on other EPF. So this goes the same with the efficacy of EPF in various weather patterns. Therefore, further research is needed to fill the gaps. So these are the list of references that I've used in this presentation. Thank you.